Our discussion here with RNA-seq alignment. And then one of the things that we want to achieve in terms of processing the raw RNA-seq data is certainly looking at the gene counts, right? We want to count these genes and get the quantification so that we know in terms of differences, which ones are related to the specific patient, which one are more related to the specific treatment, and so on and so forth, or exposure, and so on and so forth. So what you see here is giving us an illustration of the complexity of the decision in terms of mapping. So we may have, this is the green part here is a little bit blurry, but this is referring to this part is referring to the read itself. Okay, so we have the read in one part, and then I'm just trying to change the color here to okay. So this is the read. And this is a direct, we are able to get the read as substring or sequence, and then we are able to match it to the gene reference. So we can say, okay, there is one increment of that gene and keep counting the number of transcripts that we were able to map, okay, that we were able to map to this gene. So we get the quantification. So in this case, intersection strict and intersection non-empty means we are, we are fine. So this would be more clear in the other examples. For example, if I'm looking here, intersection between the number of or the nucleotides and the part of the sequence, the gene of the gene, and the ones of the transcript, I see only a portion of that read being mapped here. So it's my decision to quantify that, and it depends. This is one of the things that where we can see different scenarios here. Only a portion of the read is mapped here. So in this case, if I would go with intersection strict, meaning that, I will not accept this, then there's no feature to be recognized. And if I say, well, I'm fine with that, then I know that it's matched to gene A. And similarly, you go with these examples. So here, maybe part of it is aligned with gene A and so on and so forth, part of it with gene A and part of it with gene B. So it's ambiguous, okay? It is ambiguous in this case. So of course, anytime we want to take the read and align it to the reference, we may get some challenges. These kinds of decisions, these kind of cases, these kind of details are what make the different tools unique in terms of specifically alignment tools. So eventually we want to get to the gene expression, because expression levels are coming from microRNAs, cDNA, and then these expression levels is what we want to estimate. And so we can see the difference between gene A, gene B, and gene C. However, we need to understand if we run different libraries, there's the factor of the library. How many reads are we getting from that library or that specific experiments? So you need to know, is this gene here higher in terms of numbers because I had a library of 40 million as compared to gene B, which had library of 20 million, or both they had 40 million? And this is just a mere reflection of that expression. In other words, what I'm trying to say is we need to fix something, especially when we're looking with different libraries, experiments, protocols. Having several experiments could raise an issue of, the sequencing depth, and sometimes maybe there is some what we call batch effect. The fact that you run the same experiment again, giving everything fixed, there is still going to be a little bit of change, and sometimes it's major. you know, they have tried to examine all of these things, and always there is some variation, including, you know, running the same experiment, exactly the same protocol in a different lab, and you know, research lab, they still had some differences. Right, in the same lab, again, you still have some difference, sometimes technical thing, and so on and so forth. Also, what about the length of the transcript? So we know that larger genes would have more fragments. So these are two issues, which is, you know, the sequence depth and the size of the transcript. So there's no question but that one step should be taken over this data. What is that step as a treasure hunt question? So if I ask you, I need to do something, what is that thing? Okay, it's one single term or keyword that should be simple enough, hopefully, for you to figure out. Basically, this is normalization. So I need to normalize. Okay, and again, it depends on technology, and again, sequencing depth is something you normalize for. It's, you know, length of the transcript is another thing. Okay, because, you know, it's a longer gene, so more reads are going to line up there. This is just a reflection of the length of the gene, not because it's higher exposure in this treatment. So I need to fix that. Also, we have other cases like GC content regions. We know that some regions are either GC rich or GC poor, which would reflect eventually in what we have here. So for normalizing or having some ways to read the expression data, we can look here into what's called reads per kilobase of transcript per million. This is RPKM or FPKM. This is for the single. This is used for the single read, okay, single end read, RNA-seq, and because we have F here for fragments, this is gonna be related to the paired end where we have actually fragments. And so we have the formula of that. I'm going to give you a quick example. So we may say that, you know what, we have gene length, okay. My gene length is 2 kilobase pairs, okay, 2 kilobases, or let's say 2 kilobases, okay. Total mapped, let's say the total mapped reads, and let's say that this is for paired end, okay. Yeah, we use paired end here. What are the total map reads? Of course, they are in millions, okay, tens of millions. So we have 50 million in this case, as an example, 50 million what? Fragments, okay. So we know how we are going to read this, okay, fragments. And then what else we need to have? We need to have the reads mapped to gene, reads mapped to gene. How many reads are mapped to one gene? So we can say on average we have 1,000 reads, or actually fragments. In this case, we're looking at fragments, okay, because we're going to work with the FPKM measure. So how we do this evaluation? Well, looking at FPKM, we want to get this measure. Well, basically, we are going to use a constant, which is 10 to the power of 9, and then I'm going to multiply by the number of fragments mapped to gene. Well, yeah, could have this as 1,000. This is the number here, okay. And then I'm going to divide by the number of total fragments. So here I have the 50 million, okay. This is the 50 million, and then the gene length itself is times 2. So now you do this calculations, and you get this metric, which is again, we are counting the fragments, okay, here, and so um, um, so we are able to, then we get a number, but the way to read that number, the way we read that number is where we'd say, well, per kilobase of transcript per million. So we get this estimate, okay, given our experiment. So this is a way to, when you are generally having a data set, you have a set of experiments. So for each experiment, you can evaluate actually this number and see the differences between these experiments. For each experiment, you can consider a number of, of these measures, and then see some comparison, see how things are look like. Bottom line, we are trying to capture the number of reads, the total mapped reads, and the length of the gene in kilobases. Okay, this is what we're trying to capture in this case. Okay, so for our RNA-seq pipeline, it's going to run through some set of tools, as we illustrated this whole scenario or this workflow sort of repeatedly. Either we are going to follow the alignment-based path, where we have an access to the reference file, reference genome, or we're going to go with the alignment-free. Okay, with the alignment-free path. So there is no alignment here. It's directly jumping into quantification. So the two of the very known methods and that they were developed by the authors originally, top hat and high sat. And in fact, the recommended version nowadays is high sat 2. So high sat is successor of top hat, and high sat 2 is the successor of high sat. So if you want to do alignment-based quantification of your RNA-seq, which always, like personally, when I do this, I mean, this is my own option. I try to have both. Now, it depends again if I want to do a systematic comparison, but at least I will offer both tools and see what's going to be the output. Okay. Now, if I have a good reference genome file, why not just stick to high sat 2? Eventually, this is making the task easy. Okay. But also, if I have transcriptome file, is where I'm using Callisto for very quick quantification. And so this is very interesting. Should you make both pipelines? Okay. Or should you just go and use one of them? It's very difficult a question. You may decide to depend on the latest studies, and where I would show you one of the figures in the coming slides that's indicating a systematic comparison in terms of performance. And so once you know that that's a good enough tool, why not to go with it? Okay. And again, it's something where we have different ways to do it. So I would feel if I am in the lab, I'm the bioinformatics person, and I have teams who want to run experiments, who want to offer them the pipelines or solutions, I would at least give them both options so they can choose, okay? And I will educate them about the differences of these options, but at least I will have two options, okay? It depends, it really depends. If I end up doing and have to deal with the continuously specific type of species, maybe I will do a systematic analysis and do some comparison. And especially if I have, a benchmark or sort of feeling on what the final results should look like, maybe then I can set up a baseline, make some comparison. If that is extensive enough, then I can pick up the one which seemed very quick, like Callisto and so on and so forth. And I'm going to come to this and highlight even more about these details. So Top Hat is this alignment software that's going to map the reads considering the splice junctions. We know now these splice junctions are locations where introns have been removed, and so the exons are going to be, joined together in a specific way or another. So if we are doing alignment, we have to be careful with all the cases that we may face, like this figure, right? and even more. What we may face, we should like put examples for all of those, design an algorithm that is able to handle all of these cases. Okay. So Top Hat is this software that is able to look into these splice junctions, and so it uses a bow tie aligner to map reads to the genome, and then it analyzes the unmapped reads. So it takes two phases. We know that there are reads that we can map directly. There are ones that we are unable to map. Then we can identify what could be the possibilities, what could be the splice junctions. HiSat came later on, okay, after as a successor of, as I said, for Top Hat, and it used a hierarchical indexing system. It uses what's called a global index and a local index. So it had a global index, okay, and then a local index. Two types of indices. The global index represent the whole genome, and the local index represent small subsections of the genome. For example, for smaller regions or chromosomes. This allowed it to quickly be able to align reads, especially across splice junctions. And so Top Hat is no longer actively developed. The authors recommend using HiSat, the second version of HiSat 2, sorry, okay. And so it has improved implementation of HiSat. It's continuously being updated. And HiSat is part of a very common, HiSat 2 is a very common, or HiSat pipeline string tie, okay, and then RNA-seq 2. So this would take us from doing the alignment, making the quantification, preparing the data, and then getting the downstream analysis, basically listing the genes that are differentially expressed. So 3 programs should be sufficient to do this task. Generally, I think it is 3 software. Sometimes it could be, no, I think it is usually 3. Okay, maybe you can skip one with some post-processing, so maybe 2 or 3 software. You might have more in your pipeline because you'll be doing quality check before that, okay? Quality check. And maybe here you want to go with more machine learning and visualizations, okay, visuals after this part. So this is where it gets extended, and this is where we see some, interesting online tools or softwares to do more analysis. Moving ahead, there is Callisto. Now, Callisto is certainly very interesting. It was published in 2016 in Nature Biotechnology. And Callisto is this program that's going to use very interesting idea. Let me go just quickly through the figure and then explain the idea that Callisto is using. So this is, of course, an example of read in black. Okay, the read is in black and 3 overlapping transcripts with exonic regions. Okay, these are the transcripts that we are trying to map. And B, this is an index that is constructed by creating what's called a de Bruijn graph. So de Bruijn graph, something you can read more about, something I would explain in bioinformatics course for computer science students, is certainly very interesting data structure. So in this de Bruijn graph, we'll have graphs where nodes are made of k-mers, patterns, and then each transcript here would correspond to a colored path. Okay. Eventually, we are trying to the full path, okay, cover the transcriptome, and we use this de Bruijn graph to construct the mapping, so be able to map the whole genome. And so eventually these values of k-mers are being read and mapped, and then based on that, the quantification is done. Now, of course, there is this indexing step, but using these k-mers, patterns extracted from the transcript. But what make Callisto unique is the idea of pseudo alignment. And pseudo alignment is not concerned with the exact position of the read. Okay. It's just going to look at the transcript that it can match based on these schemas. So let me give an example. Let's say I have transcript 1, transcript 2, and transcript 3. And let's assume that transcript 1 is made of ATCGA, ATCGA, and TTCGA, and then AA, and then let's have here ATCG, okay, and then we can put just 4 T's, and then A and A, very nice. And then a transcript 3, we can have GGAT, for example, and then CGTT, and it could just have here A and A. Okay, so these are 3 transcripts. Of course, we have a read that we are trying to match, right? So in this case, the read that is coming from the sequencing machine, our read is, let's assume it is just ATCGA. So how do we do the matching? Well, Callisto is going to use pseudo alignments. Okay, so given Kmer size of 5, which is an example, a real life application, this could, okay, so, or let's call it 5 more, okay, or Kmer of length 5, different ways to read this. Well, we take this and break it down to the list of Kmers of length 5. So the Kmers coming from here, I start with, this is my first window, so I have ATCGA. And then what is my other option? I shift by 1 nucleotide, so it's going to be TCGAT. And then I shift with another one, CGATC, and then GATCG, and then ATCGA, and then TCGAA. I guess those are all the Kmers. Very well. And of course, we do the same with the second transcript. Okay, so I can list those again here: A, T, C, G, T, and then keep going: T, C, G, T, T, and so on and so forth. And we do the same thing here. Now, what Callisto does next is actually matching this directly by some techniques like hashing and so on and so forth. It makes it very quick, especially if you have an index tree and so on and so forth. So we are able to directly say that ATCGT is here, okay, through one operation of comparison. So in this case, we are going to map this read to the first transcript. So we are going to list this transcript with an increment of 1 in terms of quantification in our final table, and so on and so forth. This is the idea of pseudo alignment. We don't need to read nucleotide by nucleotide. Okay, and of course we could have, in this process where it quickly identifies the transcript without again going base by base alignment. We break it down, simplify it. It looks pretty simple, right? Pretty straightforward, I guess. This ends up to be achieving very high speeds. So see the time as compared to known, pipelines. It's top hat cufflinks because you want to do the alignment and then quantification. Callisto is directly giving you the quantification output, really saving lots of time. Okay, and in general, see there's a considerable time for alignment and quantification. And also they have done in this study some evaluation of error. I would say it's comprehensive, but they had to synthesize some data. Again, it's not that easy to come up with a way to evaluate the error, especially in these types of studies. But eventually, they achieved reasonable error as compared to other systems. So in this case, we may know that in terms of accuracy, maybe we can gain a little bit more from other systems like Biotype, for example, which is a competitor for Top Hat and so. But again, if I have many experiments, then Callisto would be a reasonable option. So that is why, and the assignment that I've designed for this module is based on Callisto. And I think it's a very good solution to work with. You'll see this in the assignment for this module. I hope you have enough instructions to follow up that on the discovery, where you can, have your environment with Conda and then install Callisto, and then you can just run it. As I told you, things need to go through 2 steps with Callisto, which is one, building the index. So you have to build the index based on the transcriptome file. So once things are ready, now someone will come and say, well, okay, Callisto, you're saying it's faster, but actually it still has to build an index. But you do this for once only. So if I say this is for the human transcriptome, I do it once, this step. And any future samples I get for a human, I'm just going to use the index. I don't need to build or do this again and again and again. So hopefully you figure out this and you run it for the group of samples that I've shared so that you get quantifications. The output that you should get by the end should be actually a list of transcripts by, as rows and columns would be, the labels of the experiments. Now I move to the very interesting section, given that we know now that we can go and design a study, go and in the laboratory have a team to prepare all of the protocol, execute that, go through a technology. Okay, now we are experts. We are sensitive to the technology part. We don't take anything for granted. We have to ask them what technology you are using. They will tell us you are not biotechnicians, you are not, the technology maker will say, yes, you are right, but we need to understand what technology you are using to know which kind of normalization we may need to do, and so on and so forth. Eventually, this is, again, as also you need to, this may inform you into what type of protocol or pipeline you would follow. And once you establish your pipeline, you move from almost big data, gigabytes of files. We used to have an experiments about 30 to 40 gigabytes for one set of experiments or one design, okay? So they had like few control, few samples with low, medium, and high dose, and so on and so forth of different treatments. Put all of that together, 30 to 40 gigabytes. I did the alignment, processing, quantification. The final file I get is barely 50 megabytes file. So we move from gigabytes to 50 megabytes, an order of magnitude or more. So that is now, I have a small file. It's this data matrix. What would I do with this? Okay, this is now going to be very interesting, which is RNA-seq data analysis. What kind of statistics I can do, what kind of analysis or visualizations I can do. This is not going to be comprehensive and by no means. This is where I can design a full course for one full semester into what you can do with data analysis, no question about it. But what I want to introduce here is few tools that, or figures that we see commonly in RNA-seq. Expression analysis would go through, in general, specific steps. We're going to look at data preparation. We have to filter some, low feature values, like these transcripts that, didn't have enough map data. Why do you keep them in your data set? They could have influence. Normalization, which normalization technique? Again, checking data quality. Every single step. You would tell me, what? We did quality check before we do the alignment. That is one thing, and doing another quality check. So you'll be doing quality check in the technology side, quality check in the lab side, quality check in raw data, quality check in data analysis. We keep doing this. Then we want to do differential expression analysis. There are too many ways to do this. Some of the linear models would be used coming up from statistics. Some of the common methods that we hear about, lemma, HR, DSEQ, VOOM, and so on, or lemma, VOOM, and so on and so forth. Those would help us to select top features. And then we can do visualizations. Why did I put visualization to be here and not parallel to this one? For one good reason. Certainly, once you have your data, you can directly jump to data visualization. Nobody can say to you that this is wrong. Feel free to do whatever you want in data analysis and data science. But the reason I put it here is because once you move from 15,000 to 20,000 mapped genes, I want to do visualizations. And with our human eyes, we are limited in terms of how much details we can capture by the end of the day. So showing me visualization for the top selected genes, maybe 100 to 200 genes, would be more meaningful, okay, to see things. So that's why we put it in this order. So filtering, we have raw FastQ reads. We go with data pre-processing. So we do quality check using FastQC. That is very important. We can run more tools like CutAdapt for adapter removal, and then trimming like Trimomatic, FastSpeed commands to trim and filter. This is one level. This is one level when we have the raw files. Next, we're going to look into normalization. And as I explained earlier, and you remember me, quote me on that, this is here a visualization of raw data as is. So this is an example for one data set. And if we go through these samples, you see some variations in each sample, meaning, you know, this is a box plot. It is a common way to visualize the data distribution. So what do I have for one sample here? I have the reads or transcripts of all genes. And so this darker vertical line here indicates the median. And this would be the 25th percentile. This is the 50th percentile. This is the 75th percentile. So which means we order the values from minimum to maximum for each experiment. And then we use this box plot to indicate what is the median. And you know how these are called the tails. So at that point, how much variations we see or outliers going beyond this. And so this is a traditional way or typical way of visualizing the data using a box plot, which is nothing but ordering the values. And then let's illustrate this with some shape. That is the box plot. The thing here is median is not standard. Median is, you know, different. Also, we see how much we shift the distribution between many of these cases, right? Like look at the tiles here. And my treasure hunt question for you here, and not too many treasure hunt questions in this lecture. And I'm trying to also finish this time, giving you a little bit more time because there is an in-class activity and there is an assignment that I want you to jump to, and very excited for you to go and work with that, okay? But here is one treasure hunt a question for you. What could be the reasons for this? What could be the reasons for this? Okay. We're talking about samples, why could end up with different outputs. Again, we're using the same technology, so we're talking about the same sequencing width. We're working with the same transcript length, right? This is our assumptions, okay? Why do we end up with some variations, okay? Think about it and share with me your answer. Or what I'm going to tell you here that this is very natural and it's almost always the case. So whenever we see something like this, we need to do normalization. And this is the output after normalization. You see, this is exactly the same data set. We apply one technique of normalization, okay? For example, scaling reads based on library size, okay? Or using other techniques. Because this is just the raw data. If you use the raw data without normalizing, so it was a bit tricky, right? These are the raw counts. I didn't say that we have done any normalization yet, but I've told you that we have the same sequencing depth, right? In terms of a technology. and we have the same gene transcript. That's the hint. Why not to normalize again, as we have discussed earlier? We have RPKM, FPKM, and other techniques. There are many techniques on how we can do normalization, like quantile normalization, for example. Let's bring things into order. There are more statistical plots to judge on the quality of the experiments and see what is the nature of the information that we have in this case. So here I'm sharing with you two examples, which is one is plotting gene expression, the variance, versus mean, okay? This is one of the very common tools to see the distribution, and we call it a mean variance plot or dispersion plot. And this is what we use after normalization. Why? Because we want to see the relationship between the mean expression level and the variance of gene expression values across all conditions and all samples. Why do we want to do this? We want to see what is the trend of data spread as expression level changes. And maybe this helps us. Okay. Typically, if we have low mean, we would wish to have low variance. Okay. This would mean genes with low mean expression levels should have low variance. Okay. In this case, why? Variance means more variability. And if I have a group of genes with low mean expression level, then I don't want them to have lots of variation of variance. But what is more sensitive in our statistical analysis is certainly this is not a meaningful area, 0 and less. Okay, we're looking more into the higher end here, this region. Certainly, this is where we want to be very careful. These are, we may end up filtering even portion of that, but this is the area where, what is happening, guys? This is the area where we have genes with meaningful abundance and counts. So as this goes from top here and start getting stable here, this means it's a nice behavior. As the mean increases, meaning those genes with higher means, the variance is low. What does it mean if they do not have had a high variance? It means what? You run one experiment with one gene, you get very low counts, you get to run the other replicates, and the same gene now give me very high counts. This is not meaningful. So if you see this figure, this plot's going up like this means there is something wrong, okay? So this is a hint for what we are trying to do. And again, we use principal component analysis, which is a dimensionality reduction technique, something from numerical linear algebra, where they give it all of the data set, and then you're going to derive two components, which are just based on assigning weights for all the dimensions you have. In other words, this is a linear combination of the variables, so we end up having samples where each point here is a reflection of all the information for all the genes expressions in this specific sample. And the same thing here of all genes and here. So why this matters? Because then I see if the control are grouping together, the medium together, and the high together. If they were all to overlap on top of each other, it's going to be, you know, imagine you spent a few $1,000 and everything turns out to be overlapping on top of each other. If you end up having this, okay, don't mind reaching out to me. I can give you hints, but do not, regret, do not stop. It might be experimental side. It might be that there is some error in the analysis in your pipeline. Maybe this is actually the data. Maybe principal component analysis is a dimensional reduction technique. Maybe there's some limitation with that. Let's refine it. So there are a few things that we can do before we say, oh, you know what? We wasted the money here. Okay, repeat the experiments. So there is our set of software packages where you can do different kinds of normalization, like scaling factors, quantile, RPKM, which we have discussed, and so on and so forth. So this is more of a reference for your own, experiments. Let me highlight here DE-seq for RNA-seq data normalization and differential expression analysis. DE-seq is certainly one common and very common option for downstream analysis in RNA-seq data. So after you have the row counts, this is what it requires. It requires the row counts. It's going to estimate some size factors, basically trying to get some median estimate and normalize these row counts, and then try to find the variance. Then make an evaluation of the parameters of the negative binomial generalized linear model. RNA-seq data is general following the negative binomial distribution. If you're interested, you can read about this, or we can discuss this more in the office hour. So once we have probability density function distribution, we can then do differential expression analysis, and so we got the results. It is basically statistics. And instead of writing some formula for you, I decided to show you this as an illustration. So I made this illustration to give you a feeling of the sequence of the steps that DE-seq2 would do. What do we get from DE-seq2 by the end of the day is p-value and log fold change. P-value and log fold change. Just one comment for negative binomial distribution. Okay, so it is more appropriate for the RNA-seq data because RNA-seq data that we get or counts are discrete non-negative integers. Okay, and especially when the variance happens to be greater than the mean, which is a common feature in RNA-seq data. Okay, now what do we see here is a very famous plot. I don't know if they ever would do this, like let's vote for the most famous plot in science. There's no question, but volcano plot is very famous. In x-axis, it has this interesting shape, and it is symmetric, okay? Because you take the log fold change, so this is basically taking the ratio of the numbers of the expression values of the treatment, average treatment over average control, and so on and so forth. So we get these distributions of these different genes. P-value on the other end is indicating me how much of significance we have here. The smaller the p-value, the more desired the outcome is to be. And so if we apply minus log 10 p-value, we are reversing this to be the higher it is. So in other words, we are very interested in this region, these set of genes, because these set of genes, they are not only significant in terms of p-value and where they show big difference between two groups, but also we can confirm this with log fold change. There is two metrics confirming this. Okay, so volcano plot is an illustration where we can visualize which set of genes showed that interesting we're more responsive in our data set, and as well as confirming that we have this behavior of the data set. Sometimes the p-value is adjusted using what's called to control what's called false discovery rate. And here there are different statistical methods. Okay, again, this is sort of material that you would get in a statistics course. Of course, as a bioinformatician, you are highly recommended to take statistics course. This is where I would love to dive into this, and again to see how we get the p-value, which is 0.05, and then we end up fixing that using, for example, some procedures, okay, where we are adjusting the p-value using the false discovery rate, which is the expected proportion of false positives among all positive findings. And here we start relating null hypothesis to rejections using, for example, Benjamini-Hochberg procedure and so on and so forth. But the point here is to tell you that when we see the results, you can expect to see p-value and the adjusted p-value. So keep an eye on both, and usually this is, not as, this could be very significant, but this is more conservative, the adjusted p-value. So sometimes you can use this for a threshold, maybe sometimes you can use this one. Which brings me to the question of what is a p-value? What is a p-value? Okay, p-value. This is very interesting discussion. Is one of the things that you can spend hours looking for resources, asking ChatGPT, looking for YouTubes, attending lectures and courses, and still you feel you are not getting it. There are different ways into how we approach this. Nor I'm going to claim I'm going to squeeze it and give you the final answer. But I want to give few hints and try to attach this to the intuition. Not to say it's complicated. In fact, it is pretty straightforward. But again, it is one of these concepts that you need to put all the components of the puzzle so that everything would make sense for you. But I'm going to black and dark the screen and tell you what is the story of this and why do we need it. Well, the idea is straightforward. I have two groups of people, okay? I have two groups of people. And please forgive me here if my drawing is not the best, okay? Now, I can relate this to bioinformatics, saying we have normal and patients, but for the time being, let's just go with, let's simplify it. I have two groups of people. Let's say here we have 5 guys, and also there we have 5 guys, okay. Okay, fine. So now, I mean, this is just the green one looks bigger because it's my own drawing, my own hand, nothing bigger in size. We actually are interested to know the differences. Are they different from each other? This is the basic question. This is the basic question. One would say, is that a good enough question? No. Why? Because what difference you are looking at? So we may look in terms of strength, we may look in terms of height, we may look in terms of weight. It depends. Why would we be interested in these questions? Well, I want to know if those two teams are going to compete in a basketball court or in a UFC arena. I want to know if it makes sense that they meet each other, okay? We need to have some similar weight, class weight, or some, equivalency giving the standards of that sport. So I want to know if they are different or not. Now, if I ask you this question as a treasure hunt question, what would you do? How would you do it? I want you really to pause here and write down your own ideas. How would you compare these two groups? And please share it with me, because this is the core of p-value. And understanding this question is going to make it very easy for us to build on this, okay? To capture the p-value concept. So, would you take just the average of one team, the average height, and compare it? Well, this is going to discard the individual features. Would you build then a pairwise comparison? Well, pairwise comparison might be reasonable, but then how I would reach at a conclusion? Because pairwise, I would certainly make the difference, compute the difference between weight of each of these. Oops, this is not nice. Okay, not nice. Had to go away. Quickly here, few guys. So if I'm trying to compute the the differences between those two groups, okay, and then of course I will end up looking at pairwise comparison, which is good, by the way. It's a good approach because I want to compare this guy to this, and this guy to this one, this guy to this one, this guy to this one. But again, when do I reach a conclusion, and how would you be able to tell me that they are different or not? Okay, here where the idea of p-value comes, which is saying the following. What about learning the distribution of this one? For two reasons. The first reason is, first, the distribution would mean I can use a sample. We don't know the whole population. What do you mean by the whole population? Well, this competition of the competition we have or the tournament we have is not one team or two teams, okay? This is to simplify it. We have 10 teams, and each team is having 4 or 5 guys. Okay, so what? Well, in the future, you'll have more and more. So you need to establish a technique or a method that is able to adapt to the idea of population. Okay, so the best way is to have a distribution or density function and adapt that to, learn the parameters based on some sample and, because we cannot keep track of every single case. And even we cannot go and have access to all the thousands of students on campus and do all these comparisons to find our best choice. We're going to take some sample and see in general what's going to be the case. So we have to be reasonable in our capacity or capability to do such test. So we end up using some probability density function. What about comparing both distributions? So if I have one distribution here, maybe it would look something like that, who knows, and another distribution here. If I want to compare these two distributions, how would I do it? Okay, so it depends on the type of distribution. These are all assumptions, guys, in the different statistical tests. You will find that there are different assumptions about the distributions. If this is a normal distribution, what are the parameters of a normal distribution? We always talk, whenever it's normal distribution, Gaussian distribution, or bell-shaped distribution, about the average and the standard deviation. So let's use the mean and standard deviation. To do that, okay, we can have a distribution here and there, and then we can come and say, you know what? I'm going to represent this group because I need to look at the average. So whoever stated their answer as the average and or pairwise is reasonable. Both are reasonable, but we need to have a more solid technique, okay? So let's say that the average of those guys is average of the green team, okay? And here is mu of the magenta team, okay, M. So I want to compare those two. Here's the strategy or interesting strategy that comes with the p-value, where we are going to establish what's called the hypothesis testing. The hypothesis testing is saying, let's go with the hypothesis and assume that both are equal, which is fine. By the way, you can assume both are unequal, because traditionally, once we are taught this content, it's always starting with equal sign. But you can assume that they are unequal, and we call this the null hypothesis, the basic hypothesis, the original hypothesis that we're gonna go with. Assume they are equal. Okay, now let's look into an interesting scenario. Okay, what is that interesting scenario that we want to work with, given what we are referring to as the null hypothesis here? We want to establish a probability measure that would tell us what is the probability of the data being at least as extreme as what we observed, given that the null hypothesis is true. In other words, or let me repeat this. What I want to try to do here is, let's assume that they are equal. But what is going to be the chance to look extreme values and find out that those extreme values that we are dealing with, or data that looks at the extreme ends of these cases, would actually be close to the observed cases? In other words, and again, this is just a story I'm trying to make. I hope it matches the definitions here and the future thing. We want to establish a probability measure. That is the p-value. P-value is the probability measure of data being at least as extreme as what we observed. This is the third time I repeat it here, but what is extreme? What is what we observed? Okay, so let's say now we have an average, which is the common case. What about now joining this with the pairwise comparison? And so look at one cases. Let's pick up two cases from here and here. Maybe they are the tallest guys. I don't know. Maybe the guys that looks the strongest. I don't know. In each team. And maybe those are what we consider the two extreme cases. So pick up those, okay? And again, it is a simplification of what we truly do in our formula. But again, it's more about the distribution. But again, pick up this case and try to observe and see. If I look into those two guys, are they actually different? Like in this team, this is the guy with the major strength. And this team, this is the guy with the major strength. So let's focus on them. I've assumed there is no difference. But when I compare them, do I see them as different or not different? Okay. Do I see them similar to each other as per my assumption or not? When we do this, and the probability is going to be very small, this is going to raise a question. Okay, the probability that to have this case and that they are actually not, turns out to be, no, they have differences. It's a big difference. I'm going to capture actually not maybe a couple of those and then a couple of those extreme cases. Maybe you have a distribution of 100 or 10 students here, or 100 students. I'm going to capture a few of them and a few from here. That would look the extreme case. And then see the average of those and the average of those. Compare. Or maybe an individual and compare. And what I'm going to see, oh, they are different. They are different. Here we go. If we start making these comparisons in a systematic way, and we see they are different, what does that mean for us, guys? It means one of two things. There are no other third scenario. It means either we are lucky, like this happened randomly, okay? We picked up those two extreme cases. Now, I'm not saying picked up two groups or two teams. We picked up extreme parts from these two teams. And I picked up these two teams, two extreme parts of these teams, sub-teams, call them sub-teams, guys, strong guys, whatever. Compare them. They are different. It could be that we are lucky, right? Actually, the teams are still the same on average. But it could be that our assumption that this, where the both averages were the same, is what? Is wrong. This is actually p-value. P-value, when it is very small, it's telling me what? There is something wrong with your assumption. There's something wrong with your assumption. Maybe they are not the same. Maybe they are not the same. Because this establishes, based on this assumption, what the chance to start seeing all of these extreme cases. And I hope this story makes some sense. And we're going to stop here with this p-value discussion. I have the formula in front of me and the type 1 error and so on and so forth. Maybe some of you are aware of it. Maybe you want to listen to this again. I hope it did not make it complicated, but I try my best to build an intuitive story of how we can build the whole picture. This is exactly where I would advise you to go again and jump to this gingerbread kind of example, which is very interesting in this link. And again, it's going to consider different distribution of these packets of gingerbread. And we know that, an example, Santa or the seller told us that I'm selling you a cookies bag of 500 grams, but we end up seeing things different. What we want to do is to use specific value. Now, we need the significance level. That is a threshold, guys, because as I told you, it could be that we were lucky, or it could be that this is actually a reflection that our null hypothesis is not valid. So we usually compare the p-value with the significance level, which is up to you to decide. Here, one of you would come and say, why on earth does Professor spend that time to explain the story? What that have to do with genes? Well, listen, guys, for genes, this is gene A, and it has distribution here for one group. And I'm going to change the color here. And we have another treatment group, for example. And I have other expression values here, okay? So I want to compare these two, right? As I did in the teams. So if I end up having 0.0005 of p-value, this means they are very different. So when I start plotting this, okay, I would see that for experiment 1, let me put this. This is my exposure 1, and let me use this here for my, oops, what happened? Pen. Okay, let's do it, okay. And then, well, the distribution is here for the values. I just put the count values as is. This count value of gene A in the first experiment, second one, third one, the replicate, sorry, of the control. I do the same thing. And I go to the other group and I plot the values. There's not even an overlap between both. This is when you expect to see p-value very small. So we have done some statistics, some analysis. We survived that part. The question now is, how can we visualize this data? What can we do with this data? Because all what we have done so far is a way to handle the raw data that was processed into this expression matrix or abundance table or count table, whatever you want to call it. And then I've done some normalization, some analysis. I've run some tool to do differential expression analysis. I know my top genes. You know, I'm hungry for visualizations. I'm personally, I don't know about you, but I love visualizations. I love to see colors. I love to see things. Even when I go to read a paper, I directly jump to the pictures. Nothing wrong with that, okay? I like to see things, how they are drawn, and then we can read more. But visualization, certainly a very important part for us human. So why to visualize RNA-seq data? It helps us with the quality assessment, as we saw in the mean variance plot. We can do what's called exploratory data analysis, which is an iterative process where you actually generate some plots, give them to your teammates, communicate the data and have discussions and reiterate on enhancing that process, getting a list of differentially expressed genes and see how we can point out that, and maybe matching to the pathway analysis. Go a higher level in terms of the analysis. Let's move and with these visualization that we have from just a list of genes. Although as good as it would seem, which is, you know, we have a list of genes now that are top differential expressed genes starting from all of these experiments. What about the decision making? We need to visualize more and work more and build more abstracts. Let's look at one example data set, exposure data set, see a sample RNA-seq data. You can check this data in the tool that I have developed earlier, which is called Ecotox Explorer. And here there's the chloropyrifos exposure study. This was part of the Ecotox chip project. They used this chemical and exposed the Japanese quail to it. Sorry, which is a hazardous to human. They had 5 controlled, 5 medium, and 5 high dose samples. Sorry, guys. Okay, so this is a typical example of the data set where we have rows of, what are those, by the way? This is a good treasure hunt question. What do you see here? Let me know. Okay, this is one of the treasure hunt questions. And then we have rows of genes, but what are those IDs? This is my question, okay? And columns of experiments. And this is one thing that you need to prepare for your analysis in the assignment. So we have sample labels. We can have, depending on the tool, we may add some metadata like what is the dose there and so on and so forth. We have gene IDs. We have the conditions as part of the metadata and the counts in this matrix. And so we want to visualize this. Now here I'm going to ask you a treasure hunt question. How do you think, based on your experience, what would be a good way to visualize this? So one of the ways that was I don't know if it invented or somebody made it. It's pretty straightforward. Define a color range, okay, color range, and then map each of these values to one part of that color range. Maybe the color range is going from green to let's choose red, okay, to red. Okay, so we define these color range can be defined as RGB values. So instead of having this value here, will say this is RGB 000, for example. 36 say, well, it's, I don't know, 20R, 6G, and so on and so forth. Have a way to normalize this and assign it to all the spectrum of this color, and then plot this. This is going to generate what's called heat maps. Heat maps, they're generally called heat maps because you can have, cold and heat, which means blue and red. So typically the example would have blue to red range of colors. This is a different color palette that I've used, which is safe for the color blindness. One of the things that we care about when we generate visualizations. Heat maps, I don't know if heat maps would win or volcano. Probably heat maps in terms of the most famous plot. Okay, it is very common in different applications, and it's nothing. Every single, this is very squeezed cell here. This is exactly this matrix exactly here. This matrix here, the full, of course, a full matrix exactly here, and again it's here. Just the numbers, but it's a way where we can see it. But as I've told you earlier, this is all genes, so it's not very interesting. We don't see the patterns. What you see here in the left is heat map without normalizing data. So if you ended up just getting something like this, dull, no information, so know that you have not performed the proper normalization. That is giving the raw data that we have. Things are very biased to the extent that we are not able to map to a meaningful range of the data. This would be the normalized case. Now, if I go and work with subset of the samples or genes, I can maybe see more information. But some of the things that we need to handle before I chop off some of the data and look at the interesting cases, I see some patterns here, but what I'm interested with is how I can group these patterns together. Why? Because I can end up with this interesting figure, where it's going to group for me the high dose cases or the control. These are the samples. Maybe this would be the control. This would be the high. Here I would see the most responsive genes and so on and so forth. So this is what we refer to in data analysis as clustering technique. How can I cluster this information together? I'm going to give you here an example, which is an animation. I think it's an interesting example where numbers, it's one-dimensional data. So we have 7, 10, 20, 38, 28, 35. This would explain and illustrate for you how we can do one type of clustering that is called hierarchical clustering. So let's put this in the number line number system. And we have 7 and 10 close to each other, and then 20 in the middle, 28 and 35. We want to group these together. So the best way is to compute distances. So let's compute the distance between 7 and 10. Well, we have the difference is 3. This is a measure of distance. Okay. You can square it. You can take absolute. You can do all these different variations of distance metrics. But this is enough for now. 10, 20, and 10, the difference 10, and so on and so forth. We have 28 and 20 is 8, 35, and 28 is 7. Okay, what do we do now? Well, we look for the smallest distance that is indicating the closest 2 points. In this case, we find 3 at the smallest distance. So I would say those should go into one cluster. So 7 and 10 would be one cluster. Now let's move on. I want to make a comparison. So I know that 20 and 28, the difference is 8. And 28, 35, the difference is 7. But what is the difference between 7, 10, and 20? This is another treasure hunt question, and I want you to write down what you think is going to be the answer. Okay, I'll give you a moment here to figure it out. Great. As you think about this, well, we can do it in different ways. We can either take 1 point or the average of 7 and 10 or one of them, and these are all different variations of the algorithm. So in this case, we can take the closest point to 20, 10 for example, and we say 20 and 10, the difference is 10. Then let's look at the smallest distance. Well, it's 7, and then we can have another cluster, 28, 35. We take the difference between 20 and 10, again, similar as we did. And what is closest to 20 as from 2835 is 28, so the difference is 8. And then we have this cluster, and here you go. And the difference between these two is 10. And finally, this is what's called hierarchical clustering. How can we visualize this? Take the number line from here to here, put the distance, and then put the height to indicate the distance. We have 3 here, 28, 35, 20, and this cluster, and here you go. This is exactly how we derive this clustering that you saw in the plot earlier. So if I take you back, this is how we can derive these dendrograms, which are bringing together the set of clusters. Okay, nice. So we can derive that and enhance our visualization by ordering which gene comes to next to which gene and which sample comes next to which sample. And now we have this for all samples, but again, let's combine everything together and use differentially expressed genes. And here you go. Now, wow, seriously, don't you feel much better? And if you want to add that as a treasure hunt question, is fine. But now it feels like I see a pattern. I'm able to read the genes. I'm able to see the samples. So we put clustering, differential expression analysis, heat map, everything together comes to this beautiful picture. So here's the final part. I'm going to go a little bit quick on this part, which is enrichment analysis. Something we discussed before. We have access to some databases like David Tool or Network Analyst. I've asked you in the assignment to go with Network Analyst. Take the set of genes or data set and do the analysis, and then go one step higher. That is, know which gene ontologies, which biological pathways or biological processes are more relevant to us. So enrichment analysis can be perceived as statistical approach to identify significantly overrepresented or underrepresented what? Gene sets, not genes. So I move from differential expression analysis of genes to significant analysis, statistical analysis of gene sets. As you would see here, I'm not listing any more genes. I'm listing gene sets, ones related to membrane region, for example, okay, and outer membrane, for example, or different as part of different cell components in this case, or different processes. Also, the type of analysis could tell us what about is overrepresented or underrepresented. You will see p-value again, because statistics, as you will see p-value in different applications. Sometimes we use network to visualize this, which is going to tell us that this pathway and this pathway, they're linked together through some common genes and so on and so forth. And then how much they are enriched. The p-value here is computed differently as compared to what we have done in the previous tests for p-value. Here we have defined sets of pathways or categories. So to make it simple for you, it's the famous example. Okay, you have urn with marble. Okay, you have this basket with marbles. Okay, marbles. You have red marbles. You have blue marbles. Okay, you have just different. Okay, and there is only one slot here where you can put your hand blindly and withdraw some of these balls. The question is, what is the probability you get a ball, a red, a red ball, or a blue ball, or other colors? That is the question. So, in other words, we have gene sets, and this is okay. Let me draw it that way, so you don't confuse yourself. Gene sets has sets with groups of genes, but we want to know which genes match with the ones that are found differentially expressed. And based on that similar analogy, we're going basically to look into, this is the full size of the balls, all number of balls, how much I'm going to withdraw. This is n, so I look into combinations. K would refer to which ones are red, n minus k, which one are green or blue, and so on and so forth. I can decide to withdraw maybe one ball or two balls or three until I get to n. That's why we have the summation. So what is the probability to do that, given that I'm going to withdraw one, one ball or two balls and so on and so forth. And this is usually how we construct this probability distribution. It's called the hypergeometric or geometric test. We end up with a p-value. Again, to do an instrument analysis, they borrow this exactly. So they map the terms. And so instead now of saying we have red marbles, we say we have category, pathway, or other significant genes. Okay. And then we have the population. And this is the overlap of the gene sets. And then we have the signature, which is the full gene set, and so on and so forth. So we have list of pathways and pathways with certain map genes. You put everything in the formula, you get your list of ranked pathways, and so on and so forth. This is some of the analysis that you can do, and once it's performed, you can also see a p-value. So we are done with this lecture. I hope you enjoyed it. What I want you to do in this in-class activity, so you still have like 15-20 minutes in this lecture, so I'm not going to record anything more. I want you really to read through this. It should be, you can do it as teams, so your project teams, you can sit down and work together. I give you all the instructions in my GitHub, so give it 15 to 20 minutes. That's it. It's an R code. So run this R code, execute it, and then do some changes to generate some heat maps. I think you will love it. And you can see how you can generate a PDF report maybe or HTML page. Play with it and then move to your in-class, to your assignment. Best of luck. Please reach out if you have any questions. Thank you so much and see you next class.